I am Mrs. Sloan, and today this is a video for my honors biology students on cellular respiration. All right, so let me make myself a little bit smaller, and I'm going to get in presentation mode. So for us, this would be chapter 7. And if you're new to watching any of my videos, what we do is down below in the descriptor of the video, I have the notes that I use, and the column column 1 you fill in, and I'll help you fill that in. And column 2, add in pictures that are helpful. Okay, and we're going to start with deep breaths, because you and I do aerobic respiration so that we can get enough ATP for what our body needs. And today we're going to talk about those processes. So that's the reason why we breathe. We need the oxygen, and we eat food so we can do cellular respiration in the powerhouse of our cells. Do you remember what the powerhouse of your cell is? Mighty mitochondria, right? But you can even do cellular respiration if you're a prokaryotic cell without any mitochondria. And so what does that reaction look like? Okay, there we go. What does that reaction look like? So here's the reaction for cellular respiration, and we actually already reviewed this, right? In chapter 6. So you can see glucose and oxygen. The glucose is going to get oxidized into CO2, and the oxygen will get reduced into water, and along the way, we're going to make some ATP. All right, so on your notes, you have overview of cellular respiration basics. It breaks down carbohydrates in order to build ATP, and then the equation I already put there for you. So when we do cellular respiration, it takes place in multiple steps. And the reason is you slowly are releasing the energy out and harvesting it in little small steps. So energy, the reactions of cellular respiration allow energy and glucose to be released slowly, Therefore, ATP is produced gradually. Now, even with oxygen, remember when we talked about the first and second laws of thermodynamics? The first law of thermodynamics says that energy cannot be created or destroyed. It can just change forms. And the second law of thermodynamics says every time it changes forms, you lose some as heat. So even with oxygen, we are only 39% efficient, right? So we're losing the rest of that as heat, or it's energy left in those molecules. So still only 35% efficient. All right, now this will give us I don't know why, let me try to make myself a little bigger, sorry. Delay of game, I'm adjusting sizes here. Okay, so let me give you an overview of cellular respiration, and this right here, and this would be aerobic respiration with oxygen, so this would be the most efficient. The inefficient way only gets us 2 ATP. So you start off with glucose, and out here in your cytoplasm of your cell, you do what's called glycolysis. It's 10 steps, 10 different enzymes, and we'll go over that in a minute. But what do you walk away with from glycolysis? You walk away with a net total of 2 ATP and then 2 reduced NADs. Now, NAD are like mules. They carry hydrogens from one molecule to another. So if this NAD right here, it's packing some heat right here, it's packing hydrogen. And if we have oxygen in aerobic respiration, we do, these NADs are going to get carried to an electron transport chain. Remember how we talked about in chapter 6 how we would create a hydrogen ion gradient and use that to make ATP? Well, here are the hydrogens we're going to use to make that hydrogen ion gradient. We're going to pass the electrons along, and we will synthesize some ATP. So that's what's going to happen with this NAD. But when we're done with glycolysis, we have pyruvate, which is half of a glucose, and it's got 3 carbon molecules. And if we have oxygen, we're going to convert it to another molecule called acetyl-CoA, and guess what? We're going to make some more NAD. So we have the ability to make more ATP. Krebs cycle, I'll talk to you about that. Again, it spits out a couple ATP, but again, more NAD. All of those NADs get brought to the electron transport chain, and also there's another courier molecule we use, and that's called FAD, and that helps us make a bunch of ATP. Now, a couple things to notice. glycolysis to pyruvate happens in the cytoplasm. The pyruvate becomes acetyl-CoA in the matrix of the mitochondria. The Krebs cycle happens in the matrix of the mitochondria. And the electron transport chain happens on the inner membrane of that mitochondria. Now, in order for us to do our electron transport chain, we need oxygen because oxygen is the final electron acceptor at the bottom of our chain. It hooks up with a couple of hydrogens and forms water. And it's the electron transport chain that gets us the bulk of our ATP. And let me explain why. Here in glycolysis, this only makes us 2 ATP, and here in Krebs cycle will only make us 2 ATP. So all the rest comes from our electron transport chain. Now, while we're breaking down this glucose, we're oxidizing it in CO2, so you can see the CO2 coming out right here. Now, if you don't have any oxygen, we'll learn that as well, then we're going to do something called fermentation. You and I take our pyruvate. If we don't have oxygen, we can't move into the mitochondria. We've got to stay in the cytoplasm. We make lactic acid, which makes our muscles sore, but plants and yeast, they will make alcohol. Okay, that's the whole chapter. I just gave it to you just now. So I'm painting that big picture, the overview of that process. So, these NADs and FADs, like I said, these are carriers. And so I wanted to show you that these temporary carriers, okay, NAD and FAD are each one, they're going to take these hydrogens from glucose, okay, and carry them to our electron transport chain. And eventually it's going to be given to oxygen in order to form water. All right. So NAD is one and FAD is another carrier. So on your notes on coenzymes, it says NAD and FAD. And so for a short time, they will hold the energy/electrons, released from the bonds of glucose. So we're taking them away from glucose. Now, I want to show you, in case you're wondering what they look like, okay? So this is NAD. You do not need to memorize this, okay? But look, it's a base and a sugar and a phosphate, this NAD. And look, here's another sugar and a phosphate. Look right here. Here's NAD when it's oxidized, and when it's a courier, there's its hydrogen right there. Okay, so that's where it carries it. That is NAD, right? There's another guy called FAD. Again, you do not need to memorize these, but in case you wanted to know what this molecule looks like, look, there's some phosphates in there, this flavin right here. Here's FAD, and here's FADH2. So the hydrogens are added on right here and right here on these two nitrogens right here. So they are both carriers. So NAD and FAD each can accept electrons temporarily to form NAD reduced and FAD reduced. They are not consumed by the process. They are recycled. Like I said, they're the mule. So they carry the electrons, and then they go and drop off the electrons. And they go back and get some more electrons, and they drop them off at our electron transport chain if we have oxygen. All right. Now, the next part I want to go over with you. Okay. Again, here's an overview. This right here, glycolysis occurs in the cytoplasm. When you're done with glycolysis, you get to walk away with a total of 2 ATP and some reduced NAD. If you have oxygen, you'll move in and do the rest of it inside the mitochondria. Now, the process of making ATP is to phosphorylate something, okay, is to add on a phosphate group. And so what I want to quickly go over with you is the ways in which you can phosphorylate something, okay? So there's basically 3 different ways to phosphorylate something. The very first one is called substrate-level phosphorylation. Now, basically, that is a handoff. It's like, I have the phosphate group. Sorry, this is a spoon for eating peanut butter because I was snacking a moment ago. My Mad Max eating peanut butter. Okay, I have a phosphate group, and now I'm handing it to you, and now you have the phosphate group. It's just a handoff. So if you look right here, I'll give you an example of that. So here's BPG. It has 2 phosphates on it. This enzyme is facilitating it giving one of its phosphates away to ADP to make ATP. So it's just literally an enzyme transferring the phosphate group. So on your notes, okay, let me go back here to this one. For substrate level phosphorylation, passing a phosphate group. Okay, you're just passing a phosphate group. All right, the second way, okay, is called oxidative phosphorylation. Now, what do you hear in that? Are you hearing oxygen? I am too. So here in cellular respiration, we're also going to learn about some oxidative phosphorylation. And this requires oxygen at the bottom of an electron transport chain. And the reason is to keep the chain flowing, somebody needs to take the low energy electrons away to keep our electrons flowing. And you can use that to make a hydrogen ion gradient like we talked about in our last chapter. So you can use that to make ATP. So oxidative phosphorylation requires oxygen because oxygen is the final electron acceptor at the bottom of an ETC. And where you would see that, an example is in your ETC of your mitochondria. All right, so take a look right here. This type of phosphorylation that you see right here in glycolysis, that's your first one, okay? That's substrate level phosphorylation. This type of phosphorylation you see in the Krebs cycle, also substrate level phosphorylation. The one I just talked to you about right here is oxidative phosphorylation, where oxygen is a part of the process to make your ATP. Okay, now there's one more, and that's photophosphorylation. So what do you hear in photophosphorylation? Light, right? And I bet you're thinking of photosynthesis, right? So photosynthesis is something that chloroplasts do to make ATP. So it's associated with another electron transport chain, but the light comes at the beginning. Now, this is an oversimplification of photosynthesis in the light reaction, but here you can see electrons from water are excited by the sun, and they go through an electron transport chain, and that's how you make your ATP. So for photophosphorylation, it's associated with light. Here, I can go back here so you can see the notes, okay? It's associated with light and chloroplasts, all right? So we have talked about the different coenzymes we're going to use, NAD and FAD. These are our mules to carry our electrons, and we've talked about 3 different types of phosphorylation. The first 2 right here, we will be using in cellular respiration that we're talking about today. All right. And so the first thing we're going to do is like fill in your overview that is underneath the steps. Aerobic respiration. Okay, this requires oxygen. Aerobic respiration requires oxygen. It is more efficient, and it generates, if you don't take into consideration the second law of thermodynamics, if you eliminate that, it generates about 36 to 38 ATP. If you do include the second law of thermodynamics, and I give you that as well, it's more like 30 to 32 ATP because you don't get a complete transfer there. All right, the steps I've listed off for you. So step 1, which I'll teach you, is glycolysis. Step 2 is the preparatory reaction. And step 3 is the Krebs cycle. Another name for that is the citric acid cycle. If you want to add to your notes, it says citric acid cycle, you can put /Krebs cycle. And then the 4th step here is the electron transport chain, which is coupled with the synthesis of ATP. Now, if you do anaerobic respiration, then you still do glycolysis because all cells do glycolysis, all cells do cellular respiration. Everybody does the first step, glycolysis. But the second step, so little letter A is glycolysis, the second step is called fermentation. And this is just one type of fermentation where you make lactic acid. So anaerobic respiration is less efficient. It only generates 2 ATP. It only generates about 2 ATP. And step 1 is glycolysis, and step 2 is fermentation. All right, so let's start with the first step, which is glycolysis. This is like the Minecraft version of glycolysis. Very, very simple, okay? In its most simplest of forms right here, you take glucose, which is a 6-carbon molecule, and you break it in half, and you break it in half into 2 pyruvic acids or 2 pyruvates, okay? So 6-carbon, broken in half into 2 3-carbons. Now, the reality of this is that takes 10 steps with 10 different enzymes, okay? Now, let's just follow the carbons for a quick minute. Do you see how you have 6 carbons? And if I have 2 pyruvic acids, 2 times 3, 6 carbons are accounted for. Look at my hydrogens. I have 12 hydrogens right here, but in pyruvate, each pyruvic acid only has 4. 2 times 4 is 8. So I lost 4 hydrogens. Now, hydrogens carry with them, right, an electron, and electrons equal energy. Who got that energy? Can you find it? It would be right here. This oxidized NAD becomes reduced. It gains those two hydrogens right here that didn't end up being in our pyruvic acid. So you get two reduced NADs. Now remember, if we have oxygen, we're going to take those NADs to our electron transport chain in our mighty mitochondria and take them to the bank of the ETC and make ourselves some ATP with it. All right, so that's comes later, but I want to point out this is like, it's our mule, it's carrying our electrons, and you can also think of it as like a check we're going to cash out later for some cash money ATP. Now, you'll also notice as a result of glycolysis that you make 2 ATP, but that's not the whole story. To get glycolysis going, to get your party started, you have to actually spend 2 ATP. You have an energy investment stage, but you'll get those 2 back and 2 more. So I spent 2, got 4 back, so I net out 2 ATP from that process. So this process of glycolysis occurs in the cytoplasm of the cell. So it occurs in the cytoplasm by all cells. What cells? All cells. Occurs in the cytoplasm by all cells. It probably evolved first, and it is a universal reaction. All cells do it. Okay? It breaks down, it oxidizes glucose through 10 steps using 10 enzymes, and it requires 2 ATP to get the party started. So that's the energy investment, 2 ATP. Now don't freak out. I'm going to show you something. Scary, scary, scary. Okay, look right here. You don't have to see all of these words. Don't even worry about it, okay? But look right here, energy investment stage. I don't know if you can see that, but it says glucose. The whole thing is glycolysis. Look, I spend an ATP, I spend another ATP, and I get ATP out. Then this dark orange part, this is the energy harvesting steps. And so you can look right here. I had to spend those 2 ATP so that I could reduce to NAD, right? Those are my mules. I took NAD and reduced it with some hydrogen and its electron, so I gave it some energy. So I have 2 NADs, and lo and behold, look, ATP, 1, 2, and 3, 4. So you spent 2 up here, you got 4 out, so you netted 2, and you walked away with 2 reduced NADs. We also walk away with 2 pyruvates, where if we have oxygen, we're going to move on into the mitochondria, right? Because we want to make even more NADs if we can, so we can go to the bank of the ETC. So look at the summary right here. Oh, sorry, this picture I want to show you again. The type of ATP making you're doing here, all of this right here in your energy harvesting, that is all substrate level phosphorylation. That is just the handoff process. All right. And then now, let me keep going here. So here's your net before and after, right? So your reactants, you put in glucose and you got out 2 pyruvates. You put in 2 oxidized NAD from glycolysis, but you walk away with 2 reduced NADs. You invested 2 ATP, but you ended up getting 4 back, so you walk away. Your net gain is 2 ATP. So on your notes, on energy harvest, you reduce 2 NADs, which will go to the electron transport chain. You can see that in parentheses. It results in the formation of 4 ATP, but you only net out 2 from the process because you had to invest 2 ATP at the very beginning. And the 2 pyruvates that you made here at the end, the 2 pyruvic acids, you're going to put those onto step number 2, which is going to be transition. Now, if you have oxygen, you're going to do transition prep. But if you don't have oxygen, you'll take these 2 pyruvates and feed them into fermentation. And we'll learn about that in just a little bit. All right, so let's look what we've done so far. We've done the glycolysis part. We've gone glucose to pyruvate. We made 2 ATP, and we made a couple of reduced NAD. And if oxygen is available, we're going to do transition prep to get us ready for our Krebs cycle. All right, so going into the mitochondria, let's just review the anatomy of it, right? Remember, there's 2 membranes, an outer membrane and an inner membrane. This inner membrane right here, these folds on it, which increase what? Surface area. Those folds are called cristae, C-R-I-S-T-A-E. Okay, the very center of the mitochondria is called a matrix, okay? And the space right here between the outer and the inner membrane, right in here that's kind of light pink, it's called the inner membrane space. So on your notes, it says inside the mitochondria, oxygen is required, okay? The mitochondrial structure is the inner membrane space, which is light pink in here, is between the outer and the inner membrane, and it's the area where we're going to concentrate our hydrogen ions, like what we learned about last chapter. We're going to concentrate a bunch of hydrogen ions right here in this light pink space. And remember, this is going to create a difference between the inner membrane space and the darker matrix, because with these hydrogen ions, right, we're going to have a difference in concentration. And you're going to put that in your notes, a difference in concentration, a difference in charge, because they're hydrogen ions, a positive charge, and pH. So concentration, charge, and pH. The matrix in the middle, right here, the darker color, that is the location of the transition preparatory reaction, step 2, and the citric acid cycle, which is step 3. Okay, and then the cristae, these folds right here on this inner membrane, the folds on the inner membrane of the mitochondria, that's the location of the ETC, capital ETC, which concentrates the hydrogen ions out here in our inner membrane space. And there's a molecule called ATP synthase. Thase means it's an enzyme. ATP synthase means we're making ATP. ATP synthase complex, and that is the membrane protein that allows hydrogen ions to move into the matrix. Okay, now I'm going to tell you more about that. Remember when we talked about Disneyland, how everybody wants to go inside, and so if you concentrate a bunch of hydrogen ions in this light pink area, they're going to want to go in, and when they go in, they're going to go through that little turnstile, the ATP synthase complex, and they're going to make some ATP. All right, good. Now your next, so here is glycolysis. Again, we went over that already. So now let's go into transition prep. So I'm going to show it to you in words, and then I'm going to actually show you the molecules because it's fairly easy to understand, okay? So here are 2 pyruvic acids from glycolysis, and what we're going to do is we're going to remove a carbon off each one of these as carbon dioxide, okay? So each one is going to lose a carbon. So you can see over here in your products 2 carbon dioxides. And what we're going to do is we're going to attach on a CoA where that CO2 was. And the reason is this is going to help us get to the next step, our Krebs cycle, to feed us into that cycle. We need this coenzyme right here. So it's going to attach on, and then it's going to be called acetyl-CoA. Now, when we do that, we're also going to reduce 2 NAD along the way. So we're going to pull some more hydrogens and electrons off. So the first thing you want to have down there on transition preparatory reaction is you want to have that it connects glycolysis to the citric acid cycle. It connects glycolysis to the citric acid cycle. And each pyruvic acid is converted to acetyl-CoA. All right. Now I'm going to show you that. Take a look right here. Okay. So here's your 2 pyruvates. So these are the 2 3-carbon molecules you made from glycolysis. They each have 3 carbons. So here's 1, here's 2, here's 3. All right. And what we're going to do is, see this heart I'm making up here, this CO2, that's going to be right here. And since I have 2 pyruvates, I'm going to have 2 CO2s. And in its place where the CO2 was, and this hydrogen is going to go hop on this NAD. We're going to make, right, give our mule some more things to carry to the electron transport chain. And this CoA is going to hop right where that CO2 is. It's just like this. So this is 2 acetyl-CoAs. We have 2 carbons left here. And this coenzyme is going to help us with our next step. And we're giving off CO2. Now, let's do some math on this, right? So our glucose molecule, right? Our glucose molecule had 6 carbons in it. And in glycolysis, we broke it in half. So now we have 2 pyruvates. Those each have 3 carbons in our glucose, right? We are now going to do this transition prep where we're going to remove a carbon off of each one. So it's like, right? You're exhaling. This CO2 right here, some of this is coming from transition prep. Now I'm down to 2 carbons, 1, 2, 3, 4, because these 2 have come off as CO2. Okay, and that is on your notes right there, right? Do you see that on your notes? Okay. All right, so I am at connects glycolysis with the citric acid cycle, and then like I said, each pyruvic acid is converted to acetyl-CoA, and you're going to get 2 reduced NADs that will go to the electron transport chain, and 2 CO2s are formed. All right, now our next step we're going to do is going to be called the citric acid cycle. And what I'm going to do right now is I'm going to break this video into 2 halves. So we're going to take a little pause right here, maybe go get a snack, and then you can come back and you can watch video 2.